So yeah, I'm here to talk about food protein nanofibrils. So a little bit of a change in direction from some of the processing that we've been hearing about. Just before I get into it, I'll just acknowledge my sources of funding from the National Sciences and Engineering Research Council, Social Sciences Humanities Research Council, the Canadian Foundation for Innovation, and from my tax, all in Canada here. And of course, I am employed by the University of British Columbia. So my background is in food chemistry and in protein biophysics. And so in our lab, we're really interested in how proteins change structure and how the structure of that protein affects their functionality. And so when I think of food proteins, I'm not really thinking about peas and meat and milk at the bulk level. I'm thinking more at the molecular level. And so today I'm going to tell you more about a particular structure called nanofibrils, or another name for these is amyloid fibrils. And so what they are, they're long threads of protein molecules. So this would be thousands and thousands of protein molecules. They're about 10 nanometers wide, 10 to 20 nanometers wide, and they can be hundreds or thousands of nanometers long. And so the interesting thing here is that protein molecules, so here's a native protein. This happens to be lysozyme from egg white, and this will come up a few times in the presentation. So here's lysozyme. Somehow this native protein that you might find in egg white, somehow it changes shape and it binds with other lysozyme molecules, and they self-assemble to form these long amyloid fibrils. So specifically what an amyloid fibril is it has a beta structured core, and these beta sheets basically, so somehow this protein goes from this sort of shape to forming a beta sheet structure, and then these beta sheets stack on each other into what we call filaments or protofilaments, and two or more protofilaments can come together and sort of wind around each other and form the mature fibril. So for example, in this case, this is composed of 2 to 3 protofilaments. Now, so an amyloid fibril or a nanofibril is a very well-organized protein aggregate. On the other end of the spectrum, because we talk about protein aggregation in different contexts, especially if we're cooking food, cooking proteins, they may aggregate. On the other end of the spectrum, we have amorphous aggregates, which are sort of just amorphous clumps of protein. They probably have some organized structure, but not nearly as repetitive and reproducible as the amyloid fibril structure. So there's a lot of interest in amyloid fibrils. One reason is because under the right conditions, it seems like any protein can form amyloid fibril. So this might be the ultimate sort of end state of any protein that exists long enough, is it might form an amyloid fibril. And the reason for this is because if we think of the thermodynamics of why a protein adopts a certain structure under a certain set of conditions, here we have a native protein. This is the energy on the y-axis. This is called the folding energy landscape. It just shows the different conformations a protein could exist in. So here's the native state. The amyloid fibril is actually the most stable form that we know of that a protein can adopt. So ultimately, proteins will turn into amyloid fibrils. Now, if we take a native protein, we can induce it to form an amyloid fibril through a number of different means, by adjusting the pH, by increasing the temperature, by applying forces to it, by applying shear to the protein. And what happens is we're basically denaturing the protein. Not completely denaturing it, but maybe partially denaturing it. So we go from a native protein, the protein might misfold, and then these misfolded states bind together and form a nucleation site, and then these nucleation sites grow into the amyloid fibrils. Now, if we heat at low pH, or if we add a protease that actually cuts the native proteins up, we can actually get the proteins being hydrolyzed, cut into peptides, and then it can be these peptides that actually assemble into the amyloid fibril. So this shows that amyloid fibrillation is sort of widespread. Historically, fibrillation was associated mainly with disease. So particular proteins in the body, if they form amyloid, that is associated with disease. So Alzheimer's, for example, involves a beta peptide. Parkinson's involves alpha-synuclein. The most famous, or maybe not the most famous, but one of the most pathological proteins is the prion protein. So when these proteins form amyloid in the body, it's generally bad news. More recently, it's become appreciated that not all amyloids are bad, actually. So some of them perform a functional, beneficial function to the organism. And examples have been found in yeast, in bacteria. Here's a cartoon here of E. coli, and these are amyloid fibrils that it produces, and it uses these to stick to things. And it's a key component of bacterial biofilm. Similarly, barnacles use amyloid fibrils for adhesion, and there's even examples in humans of catalytic amyloid proteins involved in melanin synthesis and hormone storage. So these are TEM images of hormones from humans that are actually stored in an amyloid form. Okay, so not all amyloid is pathological, and in fact, compared to the native proteins, the amyloid form of the proteins are generally totally different in their structure and functionality. And so there's a lot of interest in actually intentionally making amyloid proteins, so to engineer new functionality into the proteins. So amyloid fibrils are narrow and long, much longer than the native protein. These are thousands of nanometers long. They have high strength and elasticity, and these properties can be tuned. And they have a unique surface chemistry. So if we just think of, there are 20 different amino acids that make up a protein structure. Some are polar, some are non-polar, some are charged. So we have different unique surface chemistry. And so people have been exploring different applications for these amyloid fibrils, including just in nanotechnology in general, not necessarily specific to food, but making nanowires and biosensors and adhesives, also in drug delivery and making cell scaffolding. So for example, we have examples here of gels and films. Here's an example of lysozyme, egg white lysozyme, making a fibrillosome. So it's basically around a 10 micrometer particle where the shell is composed of lysozyme fibrils. And so there are a lot of applications that are being explored for these, and including in food. So it turns out that these amyloid fibrils are good thickening agents. They can stabilize foams and emulsions, and they could be used for encapsulation. We've seen examples of encapsulation today, and also with cell scaffolding. So for example, there's interest in growing up meat, in the petri dish, so to speak, and we need scaffolds for that, and these nanofibrils could be used for that. Okay, so in the case of food protein nanofibrils, most of the work has been done using animal proteins. So for example, milk proteins and egg white proteins, including lysozyme that I've mentioned. And so, for example, here's beta-lactoglobulin, one of the major whey proteins. So this is the native protein, and this is what it looks like when it's been converted into the amyloid fibril. And as well, here's an example of lysozyme. Now, so we can make these nice amyloid fibrils from animal proteins, but what about plant proteins? There's actually a lot of interest now, as I'm sure you're all aware, of replacing animal-based foods with plant-based foods. Now, one of the major drawbacks of plant proteins in general is that oftentimes the plant proteins don't have as good a functionality as the animal proteins that they're replacing. And people are interested in using plant proteins to replace, meat, fish, eggs, dairy, perhaps other sections of the food space. So one challenge though is how do we enhance the functionality of plant protein? So here's a typical seed storage protein. How do we get this, typical seed storage protein from say peas or soybean How do we get it to behave like all of these different products? So people have started investigating converting plant proteins into amyloid fibrils as well. And so here's some examples from soybean and kidney bean and pea. And one of the things that sort of jumps out is that these fibrils look more sort of wiggly, we call them worm-like, whereas fibrils from lysozyme and whey protein tend to be straighter and more rigid. Okay, so in our lab, we've sort of gotten into this area a couple of years ago, and so we're also looking at how can we convert plant proteins into nanofibrils. And so we're focusing on legume proteins. So for example, we've been looking at lentil, mung bean, pea, and peanut. And so basically what we do is we take these legumes, we can go buy them from the grocery store, and we just extract the proteins from them. So we get a crude extract of these proteins, as we can see in the SDS page gels here. So these are mixtures of proteins. We haven't purified them in any way. And most of these proteins are what are known as bacillins and legumins. So we know in general what their structures look like. So for example, here's bacillin from peanut and a legumin from peanut. They have similar folds. They sort of make these triangle shaped structures. So we just extract the proteins from these legumes, and then we titrate the extracts to pH 2, and we heat them to 80 degrees Celsius. And we also include some stirring because that generally makes the fibrillation go faster. So we use a fluorescence dye called THT, which is sensitive to the formation of amyloid fibrils. And so we can just measure how quickly these fibrils form. And so we can see that for peanut and pea and lentil, we've measured the kinetics. And these generally form fibrils after 10 to 20 hours. We usually let them go about 24 hours. Now we're heating them at pH 2, and so I'd mentioned the hydrolysis. So in fact, these proteins are becoming hydrolyzed as they form the amyloid fibrils. So, so by 24 hours, we can see at time 0, we have these native proteins, and then as we go along in the process, these proteins disappear, and we're just sort of left with low molecular weight peptides. So the proteins are being cut into peptides at the same time they're assembling into these amyloid fibrils. Okay, and so we wait 24 hours and then we look at them, and what we see is we sort of see two general classes of amyloid fibrils from these different plant proteins. So for the peanut, we can see that the fibrils are quite straight and long, and similarly for lentil, a lot of relatively long and straight fibrils. So we call these the non-worm-like ones, whereas for mung bean and pea, we sort of get these shorter and more wiggly looking fibrils, which we call the worm-like fibrils. So we get two sort of different phenotypes from these, from the different legume proteins. So just to summarize, we can take these legume proteins, native legume protein, heat it at 80 degrees, pH 2, and we get these different sorts of amyloid fibrils. We get this different morphology that ought to have an effect on the functionality. And so that's something that we're actively investigating right now. And we're basically interested in answering the question, can we use plant proteins to make amyloid fibrils and make gels and films and use them for encapsulation in the same way that this has been done with proteins from egg and milk, for example. So can we do this with plant proteins? So that's where we're headed. We're going to continue with these functionality studies. Now, people actually have the idea of making these fibrils and putting them in food. So it's natural to ask, is that a good idea? Are these nanostructures safe to eat? So there hasn't actually been that much study of this issue yet. One of the most relevant papers comes from this group, published in 2016, where they made protein nanofibrils from various food proteins. So from whey protein and from egg white and from some legumes. And they basically looked at cell toxicity and found that these fibrils were not toxic to these cells. So that speaks to the fact that these could be safe to eat. However, there's another very interesting and special thing about amyloid fibrils, which is that they can grow by seeding they can propagate themselves. And so this is called seeding. The self-propagation is called seeding. So we could have a solution of natively folded protein, or at least non-amyloid protein. And if we add in a little bit of amyloid fibril, what we call the seed, that will induce these monomeric proteins to form the amyloid fibrils much more quickly. So it'll just increase the rate at which they assemble into the amyloid. And so we can monitor the kinetics of this process if we add a fibril seed. And so one project that we did is we looked at this with lysozyme, because lysozyme was a very popular model protein for making amyloid, hen egg white lysozyme from chicken egg white. It just happens that humans also have lysozyme. So the human lysozyme and the chicken lysozyme, they're 58% identical. So they have this very similar folded structure. So it was reasonable to think like, well, you know, could a chicken lysozyme amyloid cause human lysozyme to form amyloid? And in fact, it can. So here's the amyloid formation of just human lysozyme on its own. And if we add egg white lysozyme, it forms the amyloid much more quickly. Now this is at pH 2, 45 degrees Celsius. What we also thought was, well, you know, if I was to ingest the egg white lysozyme fibril, could that in theory pose any danger? So the first question is, well, will it survive the stomach? So if we take this egg white lysozyme fibril and treat it with pepsin and treat it under simulated gastric fluid conditions, in fact, the fibrils do resist digestion by pepsin. So then we looked at, well, do we get cross-seeding under lower temperature conditions, like sort of more mild conditions? And so this was in simulated gastric fluid conditions as well. So we're looking for, you know, if we add this egg white amyloid to human lysozyme, do we get seeding? And out of 36 samples, just one sample showed evidence of fibril formation, whereas the rest didn't, after a period of 4 days. So we're letting this go, you know, quite a long time. So basically it says, as we get under more mild conditions, cross-seeding can happen, but it becomes more rare. And of course, it requires a long time. Now, unknown to us, there was another group working on this at the same time. They were actually asking the question, do amyloid fibrils form when you cook food? So specifically, they're looking at egg white and looking into whether these egg white proteins actually form amyloid, you know, when you cook them at home, for example. So they just basically took some eggs and boiled the egg white, and then they went through some processes of trying to extract out the amyloid protein using enzymes, proteinase K and acids. And their overall conclusion was that, yes, in fact, amyloid fibrils are formed during boiling of egg white, and they estimated 1.5 to 3% of the proteins are forming amyloid fibrils. And so here's an image. Now they're much shorter than the fibrils we get when we make, you know, them from pure protein solutions, but nonetheless they do form these short amyloid fibrils. So presumably, you know, most of us eat egg whites, and we haven't, to our knowledge, developed any amyloidosis diseases from eating those egg whites. So presumably the amyloid that we get when we consume egg white is not a major cause for concern. And so, you know, more work needs to be done before we really go and put these in food. But it does show potential that they are likely to be safe to use in food, which would be good, because this also shows that, you know, there's a lot of potential that amyloid fibrils are forming during the normal processing of protein-rich foods, particularly any process that's involving high temperature and low pH for sure, but also high pressures and shear forces could induce the fibril formation. Okay, so just to conclude there, I've told you about protein nanofibrils today, or amyloid fibrils. Historically, amyloid fibrils were known to be related to disease. Now we've learned that there are functional native amyloid fibrils, and now we're interested in sort of doing bio-inspired design of protein amyloid structures, using them in engineered devices and potentially also in food. So I just want to acknowledge the students that work in my lab. So I was at University of Georgia, if anybody is from Georgia. and then I moved to UBC a couple of years ago. And again, thanks to my funding agencies that are sponsoring this work. So, and thank you for your attention.